and I'll be speaking to you today about our flow effect transfection technology and its scalability and efficiency in non-viral delivery. At Kytopen, we strive to increase access to engineered living medicines, and in doing so, improve patients' lives. We believe this can be enabled via the end-to-end value of our flow effect technology, from discovery through process development and into the clinic. We apply the technology in two embodiments today. On the left, you see what we call our flow effect array. That's a 96-well plate configuration connected to an automatic liquid handler. This is a small volume platform that we use in collaboration with our therapeutic partners for high throughput discovery and optimization. On the right is our cGMP Flow Effect TX. This is a unit that we'll be commercializing at the end of this year. The TX can process milliliter to liters volumes at throughputs of the billions of cells per minute. At the heart of both of these devices is the flow cell, which you see there on the left. And our proprietary technology utilizes electromechanical transfection, which is a unique combination of continuous flow and electrical energy. This is different than traditional electroporation and also different from mechanical methods like squeeze. In fact, if you just flow through the cell and apply no electrical energy, you get no transfection. And if you, in theory, statically apply, so flow in, stop, apply the electricity, and then flow out, you get no electroporation, or no transfection. So it is truly a synergistic effect of the two energies that produces the better yields, cell health, and function. And I'll have a couple of data slides later that'll sort of, I'll show you that as well. So because it's the same flow cell used in both applications, the cells experience the same settings and total energy at small and large volumes, enabling seamless scale-up. Here you can see the impact of that dynamic range on both cell count and time. So again, it's truly unique for me, 30 years in sort of supplying technology to this industry, something where that same sort of environment is used at the microliter scale and then all the way up to thousands of liters. In the early development of the technology, we used readily available biological materials. So we started with T cells and GFP-labeled mRNA. Once that was optimized, we first went down a variety of cell types, and you can see them there, and then moved both to RNP and DNA. What we later found was that the way we were operating the system was actually optimized for mRNA in particular. So over the last few months, we've had a sort of a renewed focus, and we re-optimized the system for knockout of RNPs on T cells, NK cells, and CD34 positive stem cells. The results have been very compelling. And provided us with a new and broader operating window than we had in the past. We are now finalizing this work to publish it and present at some upcoming events. And we have started a similar focused approach with DNA knock-in across those same three cell types. Sorry, so the optimization efforts included about 300 plus experiments. The conditions were run on that flow effect array, so the high throughput, small volume system that I mentioned earlier. If you look at both the total viable cells at harvest and the editing efficiency on the two axes, you'll notice the line across the middle there. Apologize for that. Which we normalize for what we call a standard flow effect setting, if you will, in order to be able to do a comparison across multiple runs. And what you see is that We saw vast improved results compared to both that reference, which is probably hard to see in the X there, but basically along the line here, as well as also using standard electroporation methods that are commercially available today in the triangle. So this region here, you can see where you can get higher viability and also efficiency with a variety of different electrical energies. So this is an area that we will further investigate as we move through into DNA as well. We ran some confirmation studies, and you can see here that consistent results across this. One of the real powers of the technology and the modeling that we now have is you can make trade-offs. So if you want, in this case, higher population doublings, 2x over traditional electroporation, you can still maintain your cell viability and efficiency. So you can go in and sort of work with us and determine what's most important for you, and we can tweak the system with you to provide that capability and those results that you're looking for. So we've been quite busy over the past year. Mid-year, we did a comprehensive usability study with over 12 representatives from the industry on what we called our Gen 0 at that time. Feedback from that was that the device was too big for the bench and had some other limitations. So we took all that to heart, and we decided to take some time to rework the system that you can see there, which was first debuted at Advanced Therapy Weeks in Miami earlier this past January. We also have continued our funding efforts. We were awarded an NIH grant to look at NK cells and RNPs for a multi-year study that started this past spring. And we also closed our Series A, led by North Pond, in Q3 of this year. That has enabled us to, as you saw, do a lot of that development work on the re-optimization, as well as expand our team quite significantly. We are now at about 26 plus people or so doubled since Q4 of last year. The development timeline, so as we continue to build out over the next couple of months towards the launch at the end of the year, we are going through our verification and validation process for that control unit, the consumable, the flow cell, and our transfection buffer. We are also lining up all our documentation for submission for our GMP and FDA certification. We are open for business to do evaluations on the technology, and we'll have a handful of those new devices available this summer to do that. As well as we will begin a small pilot study with a select handful of academic institutions to begin investigating target discovery with our high throughput flow effect array. Our product development effort has been supported and enabled by our quality and regulatory path. We are building towards ISO 13485 quality management system for the end of this year, as mentioned, and that will provide us with the rigor in our supply quality oversight, get us ready for and to be able to conduct audits, and as I mentioned, to submit that drug master file in support of the GMP and clinical readiness. So next steps, we are seeking collaboration, so my contact information is there, so please feel free to reach out directly to me or see me for the rest of the show if you want to learn more and see deeper into the data, into the working of the device. So, we are providing translational peace of mind, and that comes from the quality of the device itself, the performance in terms of its efficiency and viability, the scalability and seamless translation, and the quality and regulatory package that will help you meet your needs as you work through research into the clinic. So I'd just like to acknowledge the team at Kytopen. All of this work is all on them. And special thanks to ARM for giving me the opportunity to speak today. And thank you.